Hello, my name is Luke Bennett, and I am a student in MCDB 427 Molecular Biology at the University of Michigan, and today I will be going over an experiment investigating the mechanism of intrinsic termination. This video is derived from information presented in the book Molecular Biology, 5th edition, by Weaver, and the data is from the 1999 publication by Yarnell and Roberts. I'm also going to note that intrinsic termination is also called Rho Independent Termination, but that the two names refer to the same thing. What is intrinsic termination? Well, termination refers to the end of transcription of mRNA from a DNA template. The intrinsic part comes from the fact that no proteins are found to be involved in this type of termination, so the signal for termination must be from the RNA sequence itself. Further evidence for this idea comes from the fact that all of the mRNA copies from the same DNA template end in about the same place on the DNA. The question was, how could a simple series of nucleotides end transcription? The focus of this video will be on one experiment of many that helped identify this mechanism. Before going into the experiment itself, we have to look at what the scientists already knew. They knew that transcripts that relied on intrinsic termination shared common features in their non-template strand, so these features also showed up in the mRNA as well. The first feature was an inverted repeat, which is a sequence of nucleotides where the second half is complementary to the first half and can hybridize when transcribed into mRNA, forming a hairpin stem loop. This hybridization is energetically favorable and, in theory, could exert a force on the mRNA in order to reach this lower energy state. Experimentally, hairpin stem loop formation was shown to destabilize RNA polymerase when transcription was artificially paused. The second feature was a T-rich region which partially overlapped with the inverted repeat. While being transcribed, this region created a cluster of RNA uracil DNA adenine pairs in the RNA polymerase complex. These are extremely weak pairings which make a less stable DNA-RNA binding complex and also cause RNA polymerase to pause. Based on this information, the experimenters theorized that the hybridization of the inverted repeat into a hairpin stem loop was able to disrupt the DNA-RNA binding complex, which was paused and made weak from the uracil adenine base pairs it was transcribing, when it hybridized into the hairpin stem loop. This could essentially pull the mRNA out of the RNA polymerase complex or could destabilize the transcription bubble at the active site directly. Our experimenters wanted to know if the stem loop, or merely the hybridization involved in its formation, was necessary for intrinsic termination. It wouldn't be a bad idea to pause the video now and think of how you would test this. In a loosely termed runoff assay, the experimenters made many copies of DNA and attached them to magnetic beads with streptavidin biotin binding. This DNA contained a strong promoter followed by two different intrinsic termination sites, separated by 26 nucleotides. The experimenters kept the T-rich region of these sites, but modified the inverted repeats of these termination sites depending on the sample. For each sample, they also added E. coli RNA polymerase and labeled NTPs to make labeled RNA. After some time, the samples were magnetically separated by electrophoresis, giving a pellet with the DNA plus bead and the supernatant with the free RNA polymerase and NTPs. The experimenters then ran these samples on a gel and detected RNA by size through autoradiography. The RNA polymerase complex pauses at both of the termination sites and the end of the DNA due to uracil adenine pairing. If the RNA polymerase was paused during the electrophoresis, the labeled mRNA is still attached to the DNA and goes into the pellet. If, however, termination is successful at one of these spots, then the RNA and RNA polymerase will dissociate successfully and the labeled RNA will be found in the supernatant. Just remember, pellet equals paused and supernatant equals successful termination. We can tell which site the RNA polymerase was at based on the length of the mRNA transcript. We are looking at a big gel, but don't worry. I was going to uncover the gel bit by bit, but that's not what you get as a scientist or a doctor, or in a test, so I'm giving the big boy version. It's actually very easy to interpret if we break it down into tiny little bite-sized science nuggets. Let's look at the info the figure presents. DNA. This is the DNA template they used in each sample. Delta TR2 and Delta T82 are the two names for the two termination sites, each of which have half of their inverted repeats removed, making it impossible for their RNA to self-hybridize and form a hairpin stem loop. I will explain Delta TR2 H1 when we get to it, but it was very similar. Size. At the bottom are our shortest RNA transcripts from the first termination site. They called it Delta TR2. Higher is the second termination site, Delta T82, and its unmodified version called T82. And higher still is runoff, with mRNA that are as long as the DNA itself. Supernatant or pellet. Again, pellet means the RNA polymerase was paused but transcription was never terminated, so the RNA is attached to the pelleted DNA and bead, while the supernatant means that transcription was terminated, and the RNA and RNA polymerase dissociated from the DNA and remained in solution. Oligo. To get hybridization without the stem loop, the experimenters added oligomers that were complementary to the half-inverted repeat, but only for some samples. A dash means no oligomer was added. T19 means that an oligomer complementary to the first termination site's half-inverted repeat was added. T18 was an oligomer complementary to the second termination site's half-inverted repeat, and I will talk about T19H1 when we get to it. By the way, to hybridize with the mRNA sequence, would the oligomer sequence match the sense or antisense strand of DNA? Time. This is how long they let transcription run before doing electrophoresis. So now that we know that we can extract information from this figure, what does it tell us?
So just a note about some of the upcoming slides. I realized that the way that I drew the oligomer, it seemed like it may have been hybridizing to the DNA. This is not the case. It hybridizes to the mRNA every time. I was just trying to draw it in solution, and I just happened to pick a really bad place to put that oligomer. So it's not binding to the DNA, it always binds to the mRNA. Just want to clarify that.
Let's start with our first group of lanes, 1 through 6. We see that they use the DNA where both terminators only have half of their inverted repeat sequence. We also see that they haven't added any oligomers. This tells us that no hybridization in the upstream mRNA is possible, and therefore we expect little to no termination. Is this what we see? Well, 1 and 2 are the supernatant and pellet from the same sample after 45 seconds of transcription. In every pause site, we see much more signal in the pellet lane, lane 2, than in the supernatant lane, lane 1. Lanes 3 and 4 are the supernatant and pellet from a sample with the same conditions after 90 seconds, and we see the same relationship. The same happens to a lesser degree in lanes 5 and 6, which ran for 600 seconds. Since the pellet contains all of our paused transcription complexes, this seems to indicate that these hybridization-incapable samples do not terminate transcription. You may be wondering why the bands are lighter in lane 6 than in the other pellet lanes, and why the supernatant lanes seem to gain sample in the runoff position over time. This is an artifact of the experimental setup. With longer transcription time, the RNA polymerase complexes are more likely to get through both termination sites, or in this case pause sites, and simply fall off the DNA, detaching the RNA from the DNA to appear in the supernatant. Shifting to lanes 7 through 9, we see the same setup except that they have introduced the oligomer to the first termination site, allowing hybridization at termination site 1, but not at site 2. You will note that they also only show the supernatant, with the pellet being implied from here on. Do these lanes tell us that hybridization is sufficient for termination? Well, the bright bands at the first termination site in these supernatant lanes are from all of the short RNA transcripts which were successfully separated from the DNA through termination. So yes, we notice that the second termination site has very little signal, indicating that little termination is occurring without hybridization. We also notice that we have increasing runoff transcripts as we let the transcription occur over a longer period of time, as we saw in lanes 1 through 6. Lanes 10 through 12 are virtually identical to lanes 7 through 9, except the oligomer is complementary to the half-inverted repeat of the second termination site, allowing hybridization there instead of the first termination site. As expected, we see termination at the second stop site and not the first. Why do they do the same thing at two different sites? Well, the experimenters may have wanted to test the same DNA elements, but with a different nucleotide sequence, so they notice a hybridization and not the exact sequence of nucleotides that influences termination. Lane 13 is the same setup as lanes 7 through 9, except that the oligomer has a point mutation. This is the T19H1 oligomer we were talking about before, which makes it much less likely to hybridize. We see much less termination than in lanes 7 through 9, though there is still some termination, since the oligomer can still hybridize to some degree with only one nucleotide changed. If you are understanding the experiment so far, you should be able to explain why we aren't surprised at all to see little to no termination in the second termination site, or a fairly significant signal in the runoff. If you were doing really well, you would know what this lane is a control for. Lane 14 is the same setup as lane 13. But now with the DNA transcript changed, so the first termination site is now complementary to the point mutated oligomer, restoring hybridization, but now with a different nucleotide sequence, and replacing the second termination site with a wild-type stop site, which has the full inverted repeat sequence. Since hybridization is possible at both termination sites, from the oligomer at the first site and the hairpin stem loop from the second site, we see successful termination at both sites. Lanes 15 and 16 are negative controls for the DNA sequence in lane 14, but without any oligomer. We see weak termination at the first termination site since it cannot hybridize, but strong hybridization from the second termination site since it has the hairpin stem loop. Taken all together, what do these results show us? Well, first we can clearly see that hairpin stem loops are sufficient to cause termination from lanes 13 to 16, but we can also see that hybridization to oligomers can create effective termination as well, as in lanes 7 through 12. Together, these results indicate that hybridization of upstream RNA is necessary for termination, and that the stem loop terminates because of the hybridization involved in its formation. On the other hand, what doesn't this experiment tell us? Well, since the T-rich region was always present, it tells us nothing about this, or whether the pause it is assumed to induce is necessary for termination. How might they have found information about this region if they had wanted to? Well, if they had made one DNA template with the T-rich region removed from the first termination site, they would have expected to see no pausing, so no bands of that size in the pellet, telling us that this region is required for pausing, or in the supernatant, telling us that this region is important for termination. They did not bother with these conditions, or at least didn't publish these experiments if they did them, since it had already been covered by previous literature. One thing this paper leaves unsolved is whether the hybridization terminates by pulling the mRNA out of the RNA polymerase complex or by destabilizing the transcription bubble at the active site directly. If you are looking for a great way to ensure your grasp of the material, try making 10 true or false statements about this experiment. I would recommend using the phrase blank is sufficient or necessary for blank. Getting good at this sort of thinking will put you well ahead of the game in terms of designing cogent experiments and interpreting them correctly. If you found this video helpful, look for other videos by students in MCDB 427. We put a good deal of work into each, and they should all be quite helpful.